Good morning, students. Let us start new chapter, chapter number 4, Animal Kingdom. In the earlier chapter number 3, we have studied the classification of plant kingdom. Similarly, now we are going to classify animal kingdom. Before classification, we must understand several terminologies on the basis of which we are going to classify the animal kingdom. So, basis of classification is always must. For that, let us study the several terms. When we look around, we observe different animals with different structures and forms. As over 1 million species of animals have been described till now, the need for classification becomes all the more important. The classification also helps in assigning a systematic position to newly described species. Whenever a new species is discovered, it should be getting place in some class. For that purpose, we should have a basis of classification. So, in spite of differences in structure and form of different animals, there are some fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, pattern of digestive, circulatory, or reproductive system. These features are used as the basis of animal classification, and some of them are discussed below. Levels of organization. The very first feature on which the animals are classified is how the organization of cells is present, which level is present in the organism. We have studied different levels of organization, that is cellular level, tissue level, organ system level. So, organ system level is the most complex organization in an animal. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregates, loose groups of cells. That is, they exhibit cellular level of organization. Here, each and every cell is performing all the activities required for the survival of the organism. Some division of labor occur among the cell. In coelentrates, the arrangement of cells is more complex than the cellular level. The cells performing the same function are arranged in a group known as tissue. Hence, the level of organization is tissue level organization. A still higher level of organization is organ level, in which the members of Platyhelminthes and other higher phyla, where tissues are grouped together to form organ, each specialized for a particular function. In animals like annelids, arthropods, mollusks, echinoderms and chordates, organs have associated to form functional system, each system concerned with special physiological function. This pattern is known as organ system level of organization. Organ systems in different groups of animals exhibit various patterns of complexities. For example, the digestive system in Platyhelminthes have a single opening to the outside of the body that serves as both mouth and anus, and hence it is called as incomplete digestive system. A complete digestive system has two openings, mouth as well as anus. Similarly, the circulatory system may be of two types, open type and closed type. When the blood is pumped out of the heart and the cells and tissues are directly bathed in it, then the type is open. While if the vessels occur like arteries, veins and capillaries, then the type of circulatory system is known as closed circulatory system. Next basis of classification is symmetry. Animals can be characterized on the basis of their symmetry. Sponges are mostly asymmetrical, that is, any plane that passes through the center does not divide them into equal halves. When any plane passing through the central axis of the body divides the organism into two identical halves, then it is known as radial symmetry. Here in this given figure, we can see the radial symmetry. If the given organism can be divided into equal halves by more than one plane, hence the organism is known to have radial symmetry. Coelentrates, ctenophores and echinoderms have this kind of body plan. Animals like annelids, arthropods, etc., where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. The given animal is divided into two halves by only one plane, hence known as bilaterally symmetrical. Next basis of classification is diploblastic and triploblastic organization. Animals in which the cells are arranged into two embryonic layers, an external ectoderm and an internal endoderm, are called diploblastic animals. For example, coelentrates. An undifferentiated layer, mesoglia, is present in between them. In the A diagram, we can see the purple layer, that is ectoderm, the black layer, that is endoderm, and the pink layer without cells is known as mesoglia. Those animals which have cells in their third germinal layer in mesoderm in between the mesoglia, then the known as triploblastic animals. Examples are platyhelminthes to chordates. Here in the figure we can see that the B diagram shows the third layer in between the ectoderm and endoderm, that is shown with the pink color, known as mesoderm. It is also cellular here. Next classification category is coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity which is lined by mesoderm is called coelom. Animals possessing coelom are called coelomates. Example: annelids, mollusk, arthropod, echinoderm, hemichordate, and chordate. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as a scattered pouches in between ectoderm and endoderm. Such a body cavity is known as pseudocoelom. The animals possessing them are pseudocoelomates. Example: Eschyhelminthes. The animals in which the body cavity is absent are called acoelomates. Example as Platyhelminthes. Let us see the diagrammatic representation of all three types of coelomates. First is having coelom, hence the organisms are coelomates. Second is having the mesoderm as a scattered pouches, hence known as pseudocoelomates. And third is having no cavity at all in the mesoderm, hence known as acoelomates. The next basis of classification is segmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomenon thus known as metamerism. Here is an typical annelid showing the metamerism in its whole body. Next is notochord. It is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates, and those which do not form the notochord-like structure are called non-chordates. Non-chordates ranges from porifera to echinoderms, while chordate is a separate class in the classification of animal kingdom. Classification of animal kingdom. The broad classification is based on common fundamental features as mentioned in the preceding sections what we have already studied. Now let us see the classification chart of the animal kingdom. The very first classification is done on the basis of level of organization. Animals are classified into two groups: cellular level and tissue organ or organ system level. In cellular level, mostly they are asymmetrical, acoelomates, and the phylum belongs to this is Porifera. But in tissue organ and organ system level, further classification is done on the basis of symmetry, radial and bilateral. Those who have radial symmetry are acoelomates. Phylum belongs Coelentrata and Ctenophora. While with bilateral symmetry, again one step ahead classification is there on the basis of coelom. Three divisions are there: acoelomates, pseudocoelomates and coelomates. The acoelomates are Platyhelminthes, pseudocoelomates are Eschyhelminthes, while coelomates include Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. Echinoderms sometimes have exceptions in the symmetry. Now we can see that there are total 4 phylums which are acoelomates, 1 phylum with pseudocoelomate and 6 phylums with coelomates. In the upcoming lectures, we are going to study all these phylums, all these 11 phylums from Porifera to Chordata in more detail. Till then, keep studying.